In your next lab session, you will be learning how to prepare a specimen for staining and will perform several staining techniques. The samples you prepare will be examined using the compound light microscopes used in the lab. Before beginning the exercise, it is important to understand why we stain samples in the laboratory. Stains are used to add contrast to the specimens so more detail can be seen when we observe them using the microscopes. Some images are seen on the screen that show the difference between unstained cells and those that have been stained. Two vocabulary terms you need to become familiar with that pertain to specimen preparation are smear and fixation. A smear is a sample that is introduced to the microscope slide, spread into a thin film over the surface, and then allowed to air dry. You will be preparing 4 smears in this laboratory session. Please get comfortable with preparing smears because you will continue to prepare your own slides for examination when learning more advanced staining procedures. The process known as fixation serves to kill the specimen and also fixes or preserves the morphology of cells or tissues through the staining procedure. There are 2 types of fixation used to prepare specimens for staining. Chemical fixation is accomplished by applying a chemical such as methyl alcohol or formaldehyde to the specimen. Another type of fixation is known as heat fixation. After a smear has dried, you may heat fix the sample by passing it, smear side up, back and forth through the flame of a Bunsen burner. Do not leave the slide directly over the flame of the burner. If you get the slide too hot, you will destroy the structure of the cells you are trying to observe. Heat fixation will be performed each time you are asked to fix a sample of bacteria for staining this semester. Heat fixation serves to both kill the cells as well as attach them firmly to the microscope slide. Heat fixation helps to prevent the cells from being washed off the microscope slide during your staining procedure. Staining techniques make use of different types of dyes. Dyes in solution are known as stains. Acidic dyes, also known as anionic dyes, have a negative charge. The negatively charged dye is attracted to structures or components of the cell that are positively charged or are alkaline. Examples of acidic stains include India ink, nigrosin, and eosin. Basic dyes, also known as cationic dyes, on the other hand, have a positive charge and are attracted to structures or components of the cell that are negatively charged or are acidic. Some examples of basic dyes that will be used in lab this semester are methylene blue, crystal violet, safranin, and malachite green. Simple staining is a staining technique in which a single basic stain is applied to a bacterial specimen. Bacterial cells have a large number of negatively charged proteins on their surfaces. These negatively charged proteins attract the positively charged basic dye. The result of a simple stain is that all cells on the slide will appear the same color. Simple staining allows us to observe some of the morphological characteristics of the cell, such as cell size, cell shape, and cellular arrangement. To attempt to identify the bacterial cell to the level of genus or species, we will have to learn more specific staining techniques, as well as laboratory tools that aid in assessing features other than morphology of the cell. These will be addressed as we move through the semester. You have 3 sections to the lab exercise you will be completing. In Part A, you will prepare a stained sample of cheek cells, and in Part B, you will prepare a stained blood smear. You or a lab partner will be providing the cells for these 2 exercises. Please use the proper precautions when conducting the exercises and follow the instructions provided at the beginning of the laboratory session for waste and used microscope slide disposal. In Part C, the last portion of this lab session, you will prepare 2 samples of stained bacteria. Again, use proper precautions and dispose of slides in the proper containers. In all portions of this lab, your first task is to obtain a clean, dry microscope slide and properly label it. You will be labeling your microscope slides with China pencils as seen in the top image. You will follow the procedure outlined in the NCTC Microbiology Lab Manual. The stain used in this section of the lab is methylene blue. Methylene blue is a basic dye, again meaning that the dye has a positive charge. Methylene blue is attracted to negatively charged structures within the cell, such as the nucleic acids, DNA and RNA. Methylene blue ionically bonds with the DNA and RNA and therefore remains in the cell when it is rinsed with water. Structures such as the nucleus will stain darker because of the higher concentration of DNA and RNA within those structures. While mitochondria are too small to be seen with a light microscope, they too would stain because they also contain DNA. Your expected results are seen in the images on the screen. Remember, you took the cells from yours or your neighbor's cheek, so you're looking at squamous cells. In addition to seeing squamous cells, you may also see some of the bacteria that were attached to those cells in your oral cavity. The image on the right is labeled to assist you in understanding what you will be seeing in the microscope. In Part B, you will follow the protocol outlined in the NCTC Lab Manual to prepare a stained blood smear. Please make sure you properly clean the area on your finger with an alcohol swab before using the stylet to puncture your finger. The proper procedure for spreading the blood to produce the smear is seen in the image on the screen. You will allow the blood smear to air dry and then stain it with right stain. Right stain is a combination of eosin, which is an acidic dye, and methylene blue, a basic dye, dissolved in methyl alcohol. Eosin binds to structures that are positively charged or alkaline, such as hemoglobin and eosinophil granules, and stains them a red to red-orange color. Methylene blue, on the other hand, binds to negatively charged structures such as DNA and basophilic granules and stains them dark blue. Structures that are neutral stain a pale lilac. Now note in your protocol that you will not heat fix your sample of blood prior to staining the sample with right stain. This is because the methanol in the right stain serves as a chemical fixative for the blood cells. When you have completed your prepared blood smear, your results should look similar to those seen on the screen. I want you to identify the different types of formed elements you observe on your prepared slides. In order to do so, you will need to familiarize yourself with what the different types of formed elements look like. In Part C of Lab 2, you will be required to prepare 2 stained bacterial smears. You will utilize the protocols outlined in the NCTC Microbiology Lab Manual. The stain you will be using in this procedure is crystal violet. Crystal violet is a basic stain, so it will be attracted to the negative charges on the surface of the bacterial cells. Your smears will be made using 2 different species of bacteria, Bacillus subtilis and Staphylococcus aureus. The samples will be taken from pure culture broths. Pure cultures are cultures that contain only a single species of bacteria. Mixed cultures, on the other hand, contain more than one species. Working with microorganisms is somewhat different than working with other organisms. It is different because we cannot see them with our naked eye, and they are ubiquitous. The term ubiquitous means everywhere. And trust me, microorganisms are everywhere. They are in our soil, water, air, dust, food, and they live in us and on us even when we are healthy. So when we are working with bacteria, we have to take the necessary precautions to exclude unwanted microbes from our samples. In order to keep unwanted microbes from our samples, we utilize specific aseptic techniques. An additional video will be included that discusses aseptic technique. Once you have successfully prepared your stained bacterial smears, you will need to examine them. Simple stains allow one to see the cell size, cell shape, and cellular arrangement. I would like for you to examine the smears in an attempt to determine some of these characteristics pertaining to both species. When we assess the cell size, cell shape, and cellular arrangement of bacterial cells, we are examining the morphology, or shape and structure, of the bacterial cells. You are provided a handout in the Prelab reading material that describes the various shapes and arrangements of the bacterial cells.